I can get started with the introduction now. So we're excited to have our first seminar featuring speakers from the Schmidt Futures AI and Science program. The AI and Science program aims to accelerate the use of AI and drive breakthroughs in natural science, engineering, and mathematical science by funding 160 postdocs across 9 universities who learn and apply AI methods to their research. Today's seminar is focused on AI for cancer research, where we'll have 2 talks. First, we'll have a talk from Dr. Xiaoyu Zhao on the use of AI to identify tumor protein complexes predictive of chemoresistance. And then we'll have a talk from Dr. Fatema Zohora on unraveling cancer cell communication using deep learning models on spatial transcriptomics data. So we're going to start off with a talk from Dr. Xiaoyu Zhao. She's currently a postdoctoral fellow in the Department of Medicine at UC San Diego. Her research is focused on understanding complex biological systems and human diseases by integrating genome editing technologies and AI models. She completed her PhD in cancer genomics at Stony Brook University. With that, I'll pass it over to you.
Okay. So I'm Xiaoyu. I'm a postdoc. I'm a postdoc in Dr. Trey Ediker Lab at UCSD. So today I'm very excited to share our story, how we use AI to identify protein complexes to predict drug response to chemotherapy. So as we know, an interpretable AI model is very important for cancer researchers because we not only want to know whether this model is predictive or not, and also we want to know whether the biology behind the model makes sense or not. And also we want to know whether the biology found by this model, whether we can find some new biology by this model to be potentially used for biomarker discovery or therapeutic development. So one way to make the black box neural network interpretable is to replace the fully connected neural network here with some biological informed by hierarchy, such as like protein-protein interaction network. So in 2021, our lab and the Krogan lab spent a large effort to map the protein-protein interactions in cancer cells across different cancer cell types. So published in 3 science papers. So based on the protein-protein interaction network mapped from this experimental technology, so they also did community detection and enrichment analysis for the mutation, frequently mutated across different cancers. So when they do the community detection, so by relaxing the stringency, so they can organize the protein into small protein complex, from small protein complex to a larger protein assemblies, and even larger, the bio process or organoids. So in this, by the community detection and the enrichment analysis, eventually they build a hierarchical cancer cell map called NEST system in tumors. So in this this hierarchical cancer cell map, we found many rare mutated genes. They can merge into some protein complex, and these complexes are common. Most of them are very common and frequently mutated in cancer cells. And it's very interesting for us to ask and understand how these individual genes integrate with each other to contribute to the different cancer phenotypes. So then we decide to use this hierarchical cancer cell map, NEST, to build a visible neural network for the drug response prediction. So the data set we used are from the publicly available drug screening data, including GDSC, CTRP, and CCRE. So basically, they screen the drugs. They screen over 1,000 cancer cell line across individual drug and measure the drug response. And meanwhile, they profile the genomic information of every individual cancer cell line. So to build this VNN, we used the hierarchical cell map I just mentioned. So we assigned a set of artificial neurons to each of the protein complex in this nested hierarchy cell map. So here the neurons in only the neurons in the child and parent protein complex are connected. So then we can take the genomic information of every tumor cell and use as a input, so that the genetic information in this cancer cell line can flow from the top. Where's my mouse? So the genetic information of in each cell line can flow from the top. I don't know why my mouse doesn't work, but just to focus on the hierarchy. So basically, the genetic information can flow from the top gene level to the next layer of the protein complex, and then to the next layer of protein assembly, and eventually to the root system, which represents the whole cell, and eventually output produce a predicted drug response. So the in silico activity of the neuron of each protein system then can be used to score the importance of each protein complex for the drug response prediction. So in this in this story, so we only focused on one specific group of drugs called replication stress drugs. So basically, these drugs kill cancer cells by damaging the DNA replication process or DNA damage response process, including some very classic chemotherapeutics and some well-known targeted therapy like olaparib. So we built both single task and multitask drug response model for 6 RS drugs. So as you can see here, both our VN models outperform two other pan drug model, and also the matched black box model in terms of the performance, especially in the multitask VN model. So the great performance of the multitask VM model indicates, these 6 drugs did share some common mechanism, so that they can learn some other information from each other to boost the performance, the predictive performance there. So in this hierarchy, used in our VN here, as I mentioned, the in silico activity of the neurons were used to calculate the importance of every protein system. So here I'm highlighting the most important protein complex for each of the drug response prediction. So this important protein complex include both well-known the drug mechanism and some the well-established drug mechanism, like here, the left branch, the small branch here, the DNA damage repair pathway for these drugs, and also some other novel and interesting protein mechanism for this drug response. For example, like on the very right branch, you can see some complex called RTK-JAK-STAT signaling there, which I will highlight in my following slides. So this is a kind of some novel and interesting protein complex for this drug response mechanism. So till till last slides, all these results are kind of the model result of based on the model built on the cell line drug screening data. So the next step for us is we want to know whether the VM model we build can be translated to the clinical setting for patients. So we took the genomic information of the patients treated by one of the RS drug in our model. So we chose cisplatin because this is the one of the drug with the most patient data in TCGA dataset. So we throw the genomic information of these patients into our VM model and to predict their treatment response to the cisplatin drug treatment. So as you can see here, by our model, so the patients predict to be sensitive, like to be respond very well to this cisplatin treatment. They live much longer than the patient predict to be resistant for both cervical cancer and lung squamous cancer here. So here, so after testing the model performance for the patients, so the next step is about the protein complex. So we want to test whether the protein complex identified in our VM model also can do the prediction very well. So here, as you can see, the protein complex are identified to be important in our VM model, also show much higher predicted performance in patient prediction, the patient outcome, the treatment outcome prediction compared with other protein complex, and also compared with a single genes. So here, just to show you one example highlighted in our story. So basically, this is one complex. This is called RTK-JAK-STAT. This is a single complex consisted of 21 proteins, including the very upstream, the epidermal growth factor, and a bunch of receptor tyrosine kinases, and several branches of the signaling pathway proteins, and several other regulator proteins. So the panel A are showing the protein-protein interactions identified by, in the protein-protein interaction mapping. And B is just a signaling pathway for these proteins. So we try to check the prediction performance of this protein complex in the cisplatin treatment response prediction. So we found, the patients with a very high activity of this protein complex, there tend to be have much better survival outcome than the patients with a lower activity. So also on the right panel, you can see, even though the individual alteration in this protein complex, the individual gene, they have much lower predictive power. And also, meanwhile, this individual alteration, they're really mutated in patients, which means they can only represent a few patient population, indicating they probably cannot be used as a very good biomarker for a larger population of patients. But you can see here, when you integrate these individual mutations into a protein complex, the NASD89, this protein complex start to become a very powerful predictive biomarker. And this complex, they're frequently mutated in almost half of the patients here. So means it can represent a much larger population of the patients here. here just more details about this protein complex. So I'm just showing the patients who do not respond very well, who don't respond very well to the cisplatin treatment. They tend to have like the alteration and multiple alterations in this protein complex, including the amplification in the ERBB2 and the deletion of the ERBB4 and some other interesting mutations here. Lastly, in this work, so for in the VN model, there were 41 protein complex like in total were identified to be very important for this RS drug response. So we tried to do systematic validation for this protein complex by using two different functional assays. One is genome-wide CRISPR screening. The other one is the genome-wide siRNA screening. So we found 30 of out of the 41 protein complex can be validated by at least one of the functional assay here. So here pretty much the whole story of our VN model. So basically here we build a bunch of VN, the visible neural networks, to predict the drug response of RS drugs. Constructed like guided by the cancer cell map called NEST. And here I just want to give you a showcase about, this is a really good example like for the integration of AI, biology, and the clinical application potential here. And with these VNs, we try to identify the protein complex level biomarkers. And these protein complexes, they tend to be very powerful, predictive, interpretable, and translatable biomarkers in patients there. And for future directions, for sure, is to build a better next generation precision oncology model. With the the AI models are keep evolving, and we're getting more and more data. So hopefully someday we can have a perfect drug response model for patients for the clinical application. So here is ongoing community efforts. From scientists, from people across different institutes, we're trying to build hallmarks for great predictive models of the precision oncology. So for the next generation precision model, they definitely, we hope these models should have better architecture and better interpretability and better generalizability and so on. So with that, I would like to thank. This is pretty much a collaborative work. So I really would like to thank many people, especially Asha, Trey, my mentor, and all other IDICA lab members. Also, of course, the funding resource. Yeah, we still have time. So with that, I would like to take any questions of you about this. Wait.
We have a question in the room.
Hi, super interesting. A question, how do you choose the VMN structure?
How do we choose the VNN structure?
Yeah.
So the VN structure is still a forward feeding the neural network, right? So the only difference between the VN structure and the black box is this neural network is structure is built based on the structure of a cancer cell map, a hierarchical cancer cell map. So the neural network is not a fully connected it's not fully connected anymore. It's a much sparse neural network because only the neurons, the artificial neurons in the parent and the child protein assemblies can be connected.
So I'll try to push a bit more. Generally, a biological network not to be layered, but to have skip connections or multi-layer connections. Right? So if it's genealogically diverging, maybe that is the case. I guess, how do you explore the space of networks that you choose between? Or do you just choose one and it works?
Yeah, that's a great question. So it's how to choose the the network, how to choose the hierarchy or the biological network. So first, it depends on the availability, right? So before there's some VNN, they're built on the general gene ontology and some like reactome hierarchical structure. But there was no before the science paper from our from our lab before the next cancer cell map. There was no cancer specific cancer cell map. There was no cancer specific cell map. So we think because we're trying to using a hierarchy, the biological network to predict something, some phenotype related to cancer, right? So we really hope this network can indicate the information, especially specific to cancer. So I mean, it's a great benefit to use the cancer specific nested hierarchy.
What data did you use to figure out what the nested hierarchy should be? Did you use data from a few participants, patients?
Do you want me to repeat that? I'm not sure if that was very loud. Yeah, so to repeat that, I guess the question was about like what data was used to construct this hierarchy? Was it based on a few patients' data, or is this like a long-standing hierarchy that's been developed in the research in the literature through like many years of research?
So is this question more about how to build this hierarchy?
Yeah, so like was it built from a few patients' data, or did it exist in the literature, have many years of research gone into constructing?
Okay, so yes, so from this fully connected protein network to this much smaller hierarchy, so yes, they did had some like enrichment analysis, like for the mutations frequently the alterations frequently mutated across like several cancer types. I remember it's like it's a pan cancer analysis across 13 cancer types. So they only focused on the protein system or protein complex with enriched mutations in cancer patients. This is how the nested hierarchy work. I think yeah, Mark who just helped me with the Zoom. He was the first author, the co-first author of this paper. So yeah. So
I mean,
if you guys
I don't want to hijack your presentation, but I can jump
yeah,
in.
but yeah, so I think he will be happy if you guys have more questions about the nest. So you can, I mean, I feel free to, I'm pretty sure he will say feel free to contact him.
Yeah, I will say that. How about that?
Right.
I think we have a question on Zoom. Christian, you can unmute and ask your question.
Yeah. So I was curious how you trace attribution through the network. It looked like in that previous slide we were looking at where to answer the previous question, you had whether or not a neuron was active or not active. And I was curious how you make that designation. And I'm assuming you use that to trace how like the resulting predictions are made based on certain input genetic information.
So we use several neurons for each protein complex because we expect to get multi-dimensional information for each of the protein complex. So to score the importance, I think we use we do the dimension reduction and only take the first we only take the first the component of the several dimension to score the importance of the drug response. Does
that mean that you're looking at the neuron activity at inference time or the weights themselves after it's been trained? What are you doing PCA on?
I don't think it's weights. I think we use the in silico activity itself of the neurons to then we have had a linear regression model to predict the drug response based on the in silico activity of these neurons. So then to determine based on the Pearson correlation, we can determine how this protein complex are important for this drug response.
Okay. So you're using a linear probe on top of your model's features to extract importances?
Yes.
Okay. Thank you.
Sure.
Great. Well, I hate to cut the question short, but I think it's time for us to move on to our next talk, and maybe we'll be able to get a chance for some more questions at the end. But once again, let's thank Dr. Zhao.
Thank you. Thank you, everyone.
So our next talk will be from Dr. Fatema to Zohora. Dr. Zohora is a postdoctoral researcher at the Princess Margaret Cancer Center, the University of Toronto. Her research focuses on the development of deep learning models for knowledge graphs, computer vision, and NLP with applications in single-cell analysis, proteomics, genomics, and clinical data analysis. She received her PhD in computer science from the University of Waterloo and was previously an assistant professor at Bangladesh University of Engineering and Technology. So Fatema, are you?
Yeah. So I'm sharing slide. You see this?
Yes.
I will go to the reading mode. Full screen mode. Okay. So you see my presentation, I guess. Okay. So hello everyone. I'm Fatema. I'm a Schmidt AI in Science postdoc at Princess Margaret Cancer Center, and I'm grateful to you all for giving me this opportunity to share my research with you. So today I'm going to discuss how we can unravel the cancer cell communication using deep learning models on spatial transcriptomics data. And I would like to start with some brief background, then I will be explaining the workflow of the proposed method, and surprisingly, it's also called NEST. And then I will show how we validated our model and application of our model on 2 cancer samples, lung adenocarcinoma and pancreatic ductal adenocarcinoma. And I will conclude with some future scopes. So let's first see why we are interested to study cell-cell communication in cancer biology. So in this picture, I'm showing some cancer cells in green color in middle. And they are surrounded by different type of other cells, like, for example, immune cells, B cell, T cell, macrophage, monocyte. And all these cells, they are talking to each other by sending some signals. So some of the signals are trying to kill the cancer cells, but there are many signals which are actually helping cancer cells for progression through proliferation, invasion, metastasis, and drug resistance. Therefore, if we could identify the communications which are promoting the cancer progression, then we could propose some therapeutic targets for better treatment plan. Now, what we understand by cell-cell communication. Here in this slide, we see 4 cells. They are communicating with each other. Now, this green cell is sending some protein molecules, which are called ligand, to the neighboring cells. And if the neighboring cell has a receptor, which is again some protein molecule, to receive that particular type of ligand, then this green and blue cell, they can form a bond, which we call ligand receptor pair or ligand receptor bond. And through this, they can communicate with each other. Therefore, if we want to detect the cell-cell communication in a sample, somehow we have to measure the abundance of these ligand receptor proteins. And people can use gene expressions as a proxy for ligand receptor protein. So there comes this single-cell transcriptomics, which can help to measure the ligand receptor abundance. So in single-cell RNA sequencing technology, given a tissue sample, this is dissociated into single cells, then the sequencing will give us the gene expression for all the cells. However, in this single-cell RNA sequencing technology, we actually lose the information about the position of the cells on the cell surface. But that position is very important in detecting the cell-cell communication because let's say the cells which are close to each other, they tend to communicate more than the cells which are far from each other. That's why we use spatial transcriptomics, which can also provide the spatial proximity of cells. And this spatial transcriptomics is comparatively newer and more advanced technology. So what we have, given a slice of tissue, we can have about 5,000 barcoded spots where for each spot, we know their exact location on the tissue surface. That means their XY coordinate and a set of genes along with their expressions. And each spot can have 1 to 10 cells. So in short, from spatial transcriptomics, we can have gene expression metrics for all these spots and their corresponding coordinate metrics. And we observed that we can actually convert this spatial transcriptomics data into a graph structure where all the spots, they become node or vertex in this graph. And we can insert some edge representing the relations like spatial distance, ligand receptor relation, this type of things. So using this graph structure, we should be able to find the communication between individual spots. Since this is a graph structure, that's why we are applying graph attention network, which is a deep learning model. And this one can also assign some scores to the edges. So the hypothesis is edges that are assigned higher attention score, for example, these bold connections, they will be representing the communication. Therefore, we are trying to find cell-cell communication, focusing on some domain knowledge. That means which ligand can pair up with which receptor, and also through discovering some hidden communication patterns. So since deep learning models are good at pattern recognition, that's why we decided to use this graph attention network model. And this is what we call our model NEST, cell-cell communication through neural network on spatial transcriptomics. Now, I will briefly show how the model works. Our model starts with 2 inputs. One is the spatial transcriptomics data, and the second one is some existing ligand receptor database. So there exists some manually curated ligand receptor database, which tells us like which ligand can form bond with which receptor. So having those 2 information, we do some pre-processing. For example, looking at highly expressed ligand receptor genes to know which genes are active in this particular sample, and calculating the Euclidean distance between the pair of spots so that we can make a neighborhood. Now, again, this spatial transcriptomics data, this is converted to a graph where each spot becomes a node. So to represent the node, we need some input features. We are using one-hot vector for node input feature metrics. And between each pair of node, if they're close to each other based on some neighborhood threshold distance, And there exists some ligand receptor pairs between those two spots based on the database. Then we will insert some edge like this. So to represent each edge, we will use three-dimensional edge feature vector. One attribute is their spatial distance, then the ligand receptor co-expression score, and the third one is the ID of that particular ligand receptor from the database. So this is what we have in our input graph. Then we will pass it to the main deep learning module. So here I'm using graph attention network as an encoder that will generate some embedding for the node itself and its surrounding neighboring nodes. And we are training this model using contrastive learning. That's an unsupervised way of training. So since this is contrastive, it has two branches. One branch is making a corrupted version of the input graph by doing some random permutation of those edges. And it passes this graph through the graph attention network to get some negative embedding. And on the other side, it's passing the original graph to get the positive embedding. So in one side, we are trying to minimize the probability of negative embedding coming from the true graph. And on the other side, we are trying to maximizing the probability of the positive embedding coming from the true graph. And these things can be done by maximizing the divergence between the corrupted graph and the original graph. So the purpose of doing all these things is to find a representative embedding of the given sample by detecting the edges which are necessary and sufficient to represent that sample. And we observed that the edges which are recurring patterns and have stronger relation, they're as a higher attention score. So we update the edge information with the attention score. So in the output graph, I'm showing the edges with higher attention score in dark black bold edges. So we usually keep the those edges which have a high attention score, and the rest which have low attention score, we ignore those. Then I apply a connected component finding algorithm so that we can have some disjoint components like this blue component, this green one, orange one. So what it means, each component is showing a region of strong communication among the nodes or the spores or cells in that particular region. So this is how the output will look like. Like each color is showing a subgraph or like some region of strong communication. So we can see the communication between the spots like this with label direction and strength, where strength is the attention score. And we also see the names of a particular signal or interaction using a histogram. Here, the x-axis is showing the name of the ligand receptor pair, or we can also say communications. And the y-axis is the frequency of those communication. And the color of this histogram, this is actually mapped to the component or the region from the tissue surface. For example, this FN1 RPSA, this is mostly found in the blue region and also a little bit in this red region. Therefore, we can pinpoint the location of particular communication on the tissue surface. Now, how we validate our model? So we applied our model on some well-studied biological samples, for example, human lymph node, because there are many literature on human lymph node, and we can expect some set of communications to see or like to be detected by the model. So here I'm showing this lymph node annotated as T cell zones, B follicle, germinal centers. And our model finds this set of communication in the T cell zones. So here we see it's detecting CX, CR4, CCL19, CCL21 versus CCR7, this communication in the T cell zones. And they are highly referred in existing literature because they are T cell homing signals. So our model is detecting the signals that should be detected in these particular regions. And also, if we focus on a particular communication, let's say CCL19, CCR7, we can actually pinpoint their location. So like here, I'm showing the top 5,000 highly attention scored CCL19 communication regions. As I select more stronger communication, like top 1,000 or top 500 CCL19 communication, I see they are all concentrated in the T cell zones. So this is what we expected to see. So yeah, we can say our model can also pinpoint some communication in like particular region. So we compared our model with this other two tools. One is COMOT, it's an optimal transport-based model, and then niches, which is based on some dimensionality reduction techniques. So we see that none of them were able to pinpoint the location of this communication. Niches like this one wasn't able to detect this communication at all. So these are some like qualitative validation, but we also wanted to have some quantitative validation. And for that purpose, we need synthetic data, because for the biological samples, we don't really have like labeled ground truth data or like labeled ground truth communication. So that's why we made some synthetic data setup. So based on the distance of the data points or like cell spots in the tissue, we make this like equidistant data point setup, uniformly distributed data point, then mixture of distribution of the data points. Then we apply our model without any noise in the data, then with low noise, high noise. And we observed that our model is outperforming other tools on this different type of synthetic data setup. So once we have this quantitative validation, then we applied our model on some cancer samples. So here we started with this published lung adenocarcinoma sample. So this is the tissue. We have some annotation like the cancer is in the middle region. So we don't really use the annotation in our model, but we use those for some kind of validation. So this is the next output along with the set of communication found in this sample, name of those samples and their frequency. So we are detecting bunch of TGF-beta signaling. And this was also like the original paper who published the data. They said they were also detecting TGF-beta signal. However, they couldn't identify their location on the tissue surface. And in our case, we are able to detect, and we see that these signals are happening in particularly the microenvironment region, like the region surrounding the tumor. And we also validated that by aligning the expression of those particular genes with our detection. And besides that, our model additionally detected this APOE, mainly in the cancer region, and FN1 in the lymph node region. So these two were not reported by the original paper who published the data, but we could detect that. And also if we look at the literature, we see that APOE, FN1, these communications, they promote the proliferation, migration, and lymph node metastasis in lung adenocarcinoma. Therefore, we believe that next our model is able to detect the signals which are also linked with disease severity and progression. Then we apply our model on this in-house pancreatic ductal adenocarcinoma samples. So we have these two samples from different patients, and we also have some annotation like where the tumor is and non-tumor region, and also some subtype of the cancers based on gene signature. So the first experiment was we apply our model on these two samples having the similar subtype, but coming from different patients. And we could identify a bunch of signals like GGF-beta1, LGAL, S3, PLX, and B2 on both samples. And we were expecting to see this because although they are from different patients, but they have similar subtype, so there should be some set of communication common on both samples, and our model should be able to detect those. So it was behaving the way we were expecting this model to behave. And all these signals were found to be important in this PDAC cancer context. For example, TGF-beta, this one promotes fibrosis and immune invasion in pancreatic tumor. PTPRF, this can be considered as a new therapeutic target for pancreatic cancer and so on. So besides that, we see that we can also discover some subtype-related communication pattern. For example, PLXB, PLXNB2 MET, this was found especially in the classical subtype region on both samples, along with their location. So this is a new thing that we couldn't find in existing literature. Therefore, we believe that our model can detect the communication and also it can associate the communication or ligand receptor pairs to cancer subtypes. So we are looking forward in future to apply our model on different type of cancer, like multiple patients, and let's say samples from different stages of the cancer, or like different conditions like anti-cancer drug resistant, non-resistant samples, and get the communication, compare those so that we can find some therapeutic target to help improve the patient outcome. So our model, we have uploaded the implementation in the GitHub, and for some future direction, we can extend our model by considering some intracellular signal, because right now we are looking at the outside of the cell, but many things are going on, like gene regulatory network, these things are going on inside a cell. So whether we can integrate those information into the model, and also like multimodal integration, also looking beyond the existing database, if we can discover novel ligand receptor pair through some kind of transformer model. Also, we are talking with our wet lab collaborator to see if we can do some kind of wet lab validation of our findings. So that's all. I'm grateful to my supervisor, Dr. Gregory Schwartz, and other lab mates, also collaborator from other like Princess Margaret Cancer Center and University Health Network, who all helped me to carry out this research. So thank you all, and I'd be happy to take your questions.
Are there any questions? I have a question about the resolution of this spatial data. So I think you mentioned at the beginning it was like a spatial resolution of about 10 cells. So is the model just able to detect communication like between those pixels of 10 cells?
So this particular technology is called Visium, where we have like each spot has can have 1 to 10 cells, but there is a very latest one coming, which is called Visium HD, where each spot can have 1 to 3 cells. So it will be like even higher resolution, and we also have applied our model on other technology having like single cell resolution, which is called MARFISH. So the thing is, it depends on what type of data we have. Like if we have true single cell resolution data like MARFISH, then we can apply our model to get like true single cell communication. But if the input is not true single cell resolution, like this spot based, then yes, we are getting the communication between like maybe between one cell and then 3 cells like this.
Wonderful, great. And just as a follow-up question, does the model scale well to like potentially single cell data or single cell resolution data? If you were looking at like millions of cells at a time, would the model be able to scale to that level?
So this Murphy sample I was working with, here each sample has like about 5,000. I increased that number to like, let's say 15 or 30,000. So what happened if your sample has like, as you said, million of cells, something like that, then we also have a way to split the input graph into multiple subgraph and process those in parallel so that like it can handle or scale up to that large number of cells. Because what happens here, since I'm using like deep learning model and they use GPU, so when you have millions of cells, it's accommodate all those information in the GPU. That's why we have to somehow split like all those cells into multiple region so that they can be fit into the GPU memory.
I have a general question, a very interesting talk. Can you use this model in what we say, like an active learning model or setting where, you know, if you are trying to devise some treatment where you can actively experiment with different settings so that you learn more about the communication patterns?
So by active, do you mean like something like real-time scenario?
Well, yes, I mean, but where, you know, a lot of these patterns that you obtain, I imagine that you are not 100% sure about these communication networks. So then you are going to, you know, design experiments to further probe and understand, and potentially that using some kind of, you know, treatment to see how they react to that.
So the original plan was like, once we have this model, we have this model, we worked with like our wet lab collaborators. So the original plan was the communications detected by this model. Let's say we can use some PDX model where we take the cancer cells from human and put it in the mice, let it grow so that the mice will grow cancer. And if, let's say, I have detected this PLX-NB2 signal. So if that mouse is injected something to inhibit that particular protein production, whether the outcome improves or no, something like that. We were like, in future, like we are talking with our collaborator. Yeah.
Yeah, that's exactly. I was thinking, of course, you know, I'm a computer scientist, so, but you really addressed my question, and that's very exciting. Thank you.
Yeah, thank you.
So can I have a question?
Yeah.
So I know the spatial single cell RNA-seq data provide much more information compared to the regular single cell RNA-seq. But regarding the cell-cell communication, does the communication has to be like for the cells to be physically closer to each other?
So no, like in this model, we are also looking at far cells. So what I'm doing, like we are looking at the immediate neighbors and also 3 more hops like that. So this is decided by the user or like a predefined input parameter, like how far it will look. And this is how I set a neighborhood threshold to look that much further. And it also depends on how much GPU you have, because if you like make a big threshold, like make the neighborhood bigger, it will take more GPU memory. So yeah, it also depends on that, like how much capacity you have to
I see.
For. But it is not limited to only neighbors. I mean, only like adjacent nodes. Also look further.
Okay, thank you.
Sure.
Great. Well, if we don't have any further questions, let's give our speakers another round of applause.